So we investigated the relationship of maternal infections and immune dysfunction and found associations with schizophrenia, bipolar disorder, and autism in large birth cohorts. What I'm going to talk about is I'm going to present some of our work looking at prenatal. This is a focus on prenatal infection and neuropsychiatric disorders. I also study a variety of other risk factors in epidemiologic designs for psychiatric disorders, but the focus today is on infection. But this is a kind of a broad overview slide, and this involves what we call the envirome. And this is focused on schizophrenia, but one can apply it to other disorders as well. And it kind of summarizes our overall framework for thinking about this question, which is that the idea is that in in utero insult, in interaction with susceptibility genes or other environmental exposures, and through different mechanisms, physiologic, epigenetic, maybe new mutations, leads to a cascade of events that occur from birth through childhood, adolescence, and that finally lead to adult psychosis. And I won't go through all these in detail, but all these have been identified as risk factors at different points in the life course. So this is kind of like a life course approach involving these prenatal exposures which initiate this cascade of events. And there are a variety of neural processes that underlie these abnormalities. So there have been a number of studies of risk factors for schizophrenia and other disorders. A variety of the studies that looked at infections had a number of problems, including misclassification of exposure, as well as biases. And I won't go into great detail, but a key issue with these exposures is they were often derived from big populations, such as whether a mother had an infection during a certain time in development, but in large populations rather than in individual pregnancies. And a lot of studies relied on retrospective reports of maternal infections, which tend to be not very reliable. There are also a variety of biases in terms of how cases and controls get selected, and people get lost to follow-up, and that also causes bias. So we tried to improve on these methods. So by using what's called a birth cohort study. And a birth cohort study, in this study, the idea is to identify early developmental risk factors for these outcomes. And this kind of a design is the most methodologically rigorous and, we believe, informative approach to address the question. A birth cohort, for people not as familiar, is a group of individuals born within a defined time period in a defined place and followed up for certain outcomes of interest. And there are a number of methodologic advantages to the birth cohort study. First, we could prospectively ascertain exposures during the pregnancy. In the case of our studies, we've acquired prenatal biomarkers from archived biological specimens that were acquired long before the individual got the disorder of interest, during the pregnancy of the mother. We also utilize prospectively collected obstetric and perinatal records, which aren't then reliant or which aren't affected then by someone's memory or inaccuracies in reporting. We try to minimize bias using what's called a nested case control design. This is really important for the studies I'll be telling you about. So it's a study conducted among cases and a sample of controls from the cohort that gave rise to the cases. So you have this big cohort, and then from that you identify your cases and you identify your matched controls, and you have all the same data, the same kind of data, on all your cases and all your controls. So it's not as if you're more likely to be missing data from cases or controls, which can cause problems. And then a second issue is that in most studies we have what's continuous follow-up, long-term follow-up until the outcome of interest. And if you lose people, you can adjust for that because you know who left the cohort and you often know why they left the cohort. So my first study on infections and psychiatric disorders was on prenatal rubella. And this was a small study of pregnancies, all of whom were exposed to the rubella epidemic of 1964, which was actually a worldwide epidemic. It happened everywhere, including France, happened in New York City, which is where our study subjects came from. And these are the prevalences of different of schizophrenia spectrum disorders, and 20.8% of those who were exposed had a schizophrenia spectrum disorder. So following that study, we moved on to a study in California which was another birth cohort study. And this cohort itself was developed in the 1950s and 1960s. There were 19,000 live births. Then they were all in the Kaiser Health Plan in California. And it was important to study Kaiser in the United States because we don't have a socialized health system. But Kaiser represents about 40% of the health care that was given at that time in California. It's still a very big health maintenance organization, and they kept centralized records and comprehensive databases on a variety of pregnancy-related factors and psychiatric outcomes. In this CHDS study, which is a birth cohort based on the Kaiser sample, they collected blood samples and serum on all the mothers in the cohort. And myself and my colleagues followed up these individuals for schizophrenia and for bipolar disorder. And we had 71 schizophrenia cases and 94 bipolar cases. That's here. And we gave structured interviews to all these subjects, research-based interviews. So these are some of our findings. We looked at influenza antibodies in the cases and in the matched controls in the sample, and found that for the first half of pregnancy, in the blue are the cases and in the red are the controls, and this is the prevalence of exposure to influenza. So there was much higher exposure to influenza in the mothers of the cases compared to the mothers of the matched controls, but only in the first half of the pregnancy. For the second half of pregnancy, there were no differences. And you can see here the odds ratio was 3.0, or a threefold increase in risk of schizophrenia if the mother was exposed to influenza during the pregnancy. But there's this timing effect which is interesting, although it's difficult to say with any great precision whether it really is a first half effect or if we had limited power to show an effect for the second half of pregnancy. And so another important infectious organism that we've been studying is Toxoplasmosis, a ubiquitous intracellular parasite with a seroprevalence of greater than 20%. I've heard that in France the prevalence of Toxoplasma is higher. I don't know if that's you can verify that you can verify that for me perhaps. It has a lot to do with cats because Toxoplasma goes through its entire life cycle in the cat, and if the cats are kept indoors, they have very low rates of toxo. But cats that are outdoors that eat the mice get the toxo infection from the mouse, and this is excreted into the cat litter boxes. So if the mother doesn't wear gloves or takes a protective device during the pregnancy, she's prone to get Toxoplasma. There are other sources of Toxoplasma as well that are listed here. Cats are only one source, so that they get all the blame for it. It's not fair. So, and here we measure Toxoplasma IgG antibody titers in these same maternal serum specimens from cases and controls. And we found that if the titer was moderate, that is a mid-range Toxoplasma titer, there was no difference in cases versus controls, whereas for high titer, there was a much higher prevalence, frequency of exposure in the mothers of cases compared to controls, and about a 2.6-fold odds ratio, which is quite significant. We've moved on then to look at cytokines, which are a family of soluble polypeptides. And these are important in their relationships to infection because they're stimulated by a broad array of infections. So they kind of give us maybe possibly a final common pathway mechanistically to why different infections might increase risk of psychiatric disorders. But they're also increased by a variety of other factors that are non-infectious. And it's been known, we studied these particular cytokines because it's been known that they've been associated with other developmental brain abnormalities. This is before we had found these associations with schizophrenia. So in the study that I'll be telling you, we looked at 4 pro-inflammatory cytokines, interleukin 6, interleukin 8, TNF-alpha, and interleukin 1 beta. So we found for interleukin 8 that the mothers of the cases compared to mothers of controls had markedly higher levels of this cytokine. But we didn't see that for these other cytokines, at least significantly different. But the differences in the sample sizes weren't very large. So IL-6 would be one where I thought we would have seen an effect because certain animal models have suggested that, but we didn't see it here. And you can calculate this thing called population attributable fraction, which is an epidemiologic measure. And really what it tells you is how much of the disease could be potentially eliminated if you could get rid of all of whatever it is you're studying, whatever that risk factor is that you're studying. If you eliminate the entire risk factor, how much of schizophrenia in this case could you eliminate from the population? So we found, based on our data, that if you could, say, vaccinate all the pregnant women and they were responsive to it 100%, that if you could get rid of influenza infection, 14% of the cases could be eliminated. High titer toxoplasma, 13%, and there are a variety of preventive measures for that. And periconceptional genital reproductive infection, which I didn't present data on, but which we also showed an association, another 6%. So this is about a third of all cases, assuming you can get rid of all these infections, which is probably not feasible. But interestingly, though, this has marked implications for the prevention of schizophrenia. In other words, without understanding any of the mechanisms, we could potentially prevent these disorders by eliminating these infections. And then it gets to genes and germs. This is a little dated list of genes, but now there are new genes, but all of them have, well, even the new genes, they hardly ever tell you what the real effect sizes are. They show you a p-value, but they say, they don't say how much is the risk increased if you have that genetic mutation. So except for the copy number variants, which are a class of variants that probably represent a fairly small percentage of the total genetic liability to schizophrenia, except for those, most of the genes, the common polymorphisms, don't account for very much individually of the disease. It's only in combination that you see the effect. So the point of this slide really is to say that these environmental exposures, the odds ratios are much higher than for these common genetic polymorphisms. And it's polymorphisms in the major histocompatibility complex, a set of genes related to proteins which are involved in the immune system functioning, which have been among the best replicated of the genetic findings in schizophrenia. And here again, you don't see very big effects, but this finding came out, it wasn't hypothesized a priori, it came out of a genome-wide association study. So it's quite interesting in terms of the idea of an infectious or an inflammatory etiology for schizophrenia. And the MHC family is a large gene family. An interesting thing about them is that they're not only involved in the immune response, but Carla Schatz and others, people working in labs today, have shown effects on synaptic development and plasticity. And I'm not going to go through all the details on this slide unless there are questions. But it's these excitatory synapses and GABAergic synapses that have been linked strongly to schizophrenia that are affected by MHC. And it's not to say that schizophrenia is a synaptic disease, but synaptic abnormalities have definitely been suggested by the literature, postmortem studies, and a variety of other studies. So it's not just a coincidence, I think, that we found that MHC is associated with schizophrenia in genetic studies, and that's related to the immune system. The idea is that there may be true effects on brain development as a result of these infections. So this is in an epidemiologic study. We looked at, schizophrenia is considered to be the outcome. But we were interested in a complementary approach where we could look at how infection affects brain abnormalities that are found in people who have schizophrenia. So it's looking underneath, and not just what's going on, what you see, but what's going on in the brain. And so here what we did is we took our same sample that I just presented to you, and we divided them up into whether or not the mothers were exposed or unexposed to an infection in pregnancy. And we did neuropsychological testing on the offspring. They're grown-up offspring. They weren't children. We tested them. So these are schizophrenia cases, and we did it also in controls. I'm only showing you data now on cases. And we found that if the mother was exposed to an infection in pregnancy, there were many more errors on the Wisconsin card sorting test, which is a pretty commonly used test of executive functioning. And also they did more poorly on the trail-making test, which tests a variety of things, but it's also an executive functioning measure test. The trails B is, as well as a figure of fluency test. So again, this means, the reason this is higher in the exposed than the unexposed tells you that it took more time for people who were schizophrenia cases who were exposed to infection to complete the task. So it's a worse performance. And the figure of fluency test is the total number of correct. So that was worse in those who were exposed and unexposed. So all these findings are consistent with some executive function deficit in those who are exposed to infection. And then we're also able to look at whether interleukin-8, which we associated with schizophrenia in the epidemiologic study, was related to brain abnormalities by MRI. And in this slide, we looked at a very simple measure, but one of the best replicated in schizophrenia, which is ventricular CSF volume. So the higher the interleukin-8 level was, the greater the CSF volume was. And it's a nice linear relationship between the two. That doesn't mean it explains all of increased CSF volume, but the idea is that inflammation in pregnancy could be one factor, one reason as to why people with schizophrenia have larger ventricles. So we've looked across different disorders, though, not only schizophrenia, and it really goes back to the Kraepelinian dichotomy of psychosis. And Kraepelin was the first to differentiate what he called dementia praecox, which we today call schizophrenia, from manic depressive psychosis, which we still call as that, but we also call it bipolar disorder. And the issue was Kraepelin himself was perplexed about what the whether they were different or similar etiologies, and Dr. Le Boyer is very interested in this question as well. So we looked here at maternal exposure to influenza and risk of bipolar disorder. In this case, it's with psychotic features in the same birth cohort that I just told you about where we studied schizophrenia. And interestingly, now, using a clinical diagnosis of influenza given by doctors in Kaiser, mostly obstetricians, we found that the prevalence of influenza was about 5 to 6-fold higher in the mothers of cases compared to the mothers of the controls. So this is a clinical diagnosis, but then we also tested the serum for influenza antibody and found basically the same thing. Here we have about a 5-fold increase in risk. And we've done various analyses to try to correlate these with one another. They do correlate pretty well. If they're clinically diagnosed, usually we see the antibody as well. But these studies were actually done separately from one another, and they came to the same result. So what it tells us is that maybe influenza is a risk factor not only for schizophrenia, but also bipolar disorder. That doesn't mean it's infection is a risk factor for both that any infection are risk factors for both schizophrenia and bipolar disorder. So this was published in a special issue that Marion invited me to work on in Schizophrenia Bulletin, where we compared and contrasted schizophrenia and bipolar disorder with regards to various infections and looked at the data across different studies. And so what we see here is that we're the only one to show flu and bipolar. But for Toxoplasma gondii, there have been now 2 replications of our finding in Denmark and in Sweden. In this CPP study, they didn't seem to show a schizophrenia relation, but they did show a relationship with bipolar disorder and toxoplasma. And then herpes simplex virus type 2, we found no association. Other studies have found increases, which are very mild. And there's one study looking at bipolar and HSV and didn't show an association. So what's really unclear is, to us, and it's an ongoing question, is how specific are these infections are for different psychiatric outcomes. And we don't really have enough data yet on bipolar disorder to draw definitive conclusions on different infections. So we're actively studying that now in our cohorts. So and another way to address specificity is, well, if you really want to address it, you want to have samples where you have a variety of different outcomes. One of the other limitations of our work is the sample sizes are very small. So I identified basically a national birth cohort in Finland called the Finnish Maternity Cohort. In Finland, they've been drawing blood samples and stirring sera for the entire pregnant population since 1983. They draw the serum and test them to test for things like HIV and hepatitis, syphilis, things that aren't very common in that population. And we're not that interested in those things because they're not common. But the nice thing is they save all the serum that they didn't test for, and they put all the serum in one freezer. So there are 1.5 million pregnancies with serum samples. They're mostly first and second trimester. And we were able then, of course, using national registries, which are very, very good throughout Scandinavia, including Finland, For things like hospital and outpatient discharge diagnoses, psychiatric disorders, and they have a variety. They have all the other disorders as well in these registries, all the other medical disorders. Birth registers, we've used longitudinal developmental data. They have population registries, so you can do linkages between family members. I'm not going to show you all those data. And there's these called PIN codes, so you can link people across different registries with the PIN codes. And PIN codes are personal identification numbers used to identify individuals. They're unique for every individual. So basically our design advantage is now, instead of Kaiser in California, which is this health plan in one part of California, now the cohort is the whole country. And we have virtually complete ascertainment of all cases, since almost everybody gets their care through the national insurance system in Finland. And from this, we were able to get very high statistical power. We have 4,000 cases of autism. We call that study the FIPS-A. FIPS is Finnish Prenatal Studies. So FIPS-A, the schizophrenia studies, FIPS-S. We have 1,500 cases, but we expect that to double because we did our last follow-up in 2009. And then FIPS-B is bipolar, where we have about 1,900 cases. So we can look at a lot of interesting questions with these large data sets. Diagnostic specificity we can do. We can minimize selection biases because we have almost all the pregnancies and almost all the cases. So the problem with bias is if you get only some of your pregnancies and some of your cases, and you're missing differential ascertainment between cases and controls, all these things can lead to biases, which can give you false positive findings frequently. So we're able to minimize that using this design. And we've done a variety of other studies that I won't be able to have time to show you, where we have data on the developmental course of these individuals, like head circumference measures longitudinally in infancy and in childhood, and other height and weight. And so we've been working on those data, and we've published some of those data as well. But for now, I'll be focusing on a few studies that we've done from the cohort relating to inflammation. So C-reactive protein is an acute phase reactant. It's an established marker of low-grade inflammation from both infectious and non-infectious exposures, and it's substantially elevated during acute infection. Infection isn't the only cause of elevated CRP in the blood. You can have an inflammatory disorder or tissue injury. These lead to pro-inflammatory cytokines to be secreted, like IL-1 beta, IL-6, and TNF-alpha, which stimulates the liver to produce CRP. So in this study, we looked at CRP levels in the maternal serum in Finland, and schizophrenia. We had 777 matched case control pairs, so a much larger sample than in our previous study. This is showing CRP expresses a continuous variable. So this is the effect of every 1 unit increase, and it's in milligrams per liter, so every 1 milligram per liter increase in CRP on schizophrenia risk. And the unadjusted and adjusted odds ratios are here. So it doesn't look very big, but it's for every 1 unit increase. So here are the medians for the cases and the medians for the controls to give you an idea of what kinds of ranges we're talking about. And this shows a distribution of CRP values for all the schizophrenia cases and controls, which gives you a little bit of a better handle on what's going on. So the red are cases and the blue are controls. And you can see here an overrepresentation of higher CRP levels. These are the higher CRP levels and the lower levels. Higher representation of cases here and higher representation of controls here. It seems to be a rather continuous effect. It doesn't seem to be present for a certain subgroup at the high end of the distribution. We did the same thing then for bipolar disorder in this cohort. Here we had a smaller number of case control pairs, but really saw no effect. So 0.92 is an odds ratio less than 1, means there's no effect. The median for cases was actually lower than the median for the controls. And that's a distribution there. I won't belabor the point here. You can make some arguments that there might be, higher CRP here for certain subsamples, but basically does not seem to be a difference between the groups. And then we did the same thing for autism. So again, we had the same exposure, but 3 different outcomes. So in our third outcome, autism, we did see an effect. And here, again, it's the odds ratio every 1 unit of CRP. How much did that increase the risk of autism? And these effects are significant. We then looked at it in quintiles here, and we found that for the highest quintile, a significant overrepresentation of cases compared to controls, and the opposite for the low end of the CRP distribution. So 3 disorders, 2, schizophrenia and autism, seem to be related to mother's CRP, maternal inflammation. And there have been a variety of other studies that have looked at markers of inflammation, biomarkers such as interferon gamma, TNF-alpha, and others that have suggested associations as well. Those studies didn't look at CRP, but CRP has been correlated with other cytokines. There was a study by a Danish cohort showing viral infection in the first trimester and bacterial infection in the second trimester were related to autism in the offspring. Fever has been associated. A Danish cohort also showed influenza by self-report had a borderline significant association. So it's not using antibody or very good measures, but there's a suggestion, though, that influenza may be involved, and we're trying to study that now in our finished sample. And then in adulthood, there is evidence from, it's too much to summarize here, but increased microglial activity in cortical samples. These are activated macrophages that are in the brain and suggestive of brain inflammation, as well as increases in cytokines in people with autism. And some of these studies were done in children, others in adults. So we also are interested in translational research, that is animal models of prenatal infection. And there have been a variety of studies, and there are too many to describe here, that have been done so far looking at immune activation. So basically they give a drug that stimulates the immune system of a pregnant mouse or rat. There's a few studies done on monkeys, but mainly in the mouse and mainly in rodents. And these stimulate the secretion of these cytokines that I've been talking about. And they then look at the offspring after they're born. They look at the offspring for a variety of behavioral and cognitive and pharmacologic abnormalities, and basically have found a number of abnormalities that one could say are analogous to those that are found in disorders such as schizophrenia and possibly also autism. And so what we were interested in doing was looking at this. No one had really studied neural circuitry in the offspring as a result of prenatal immune activation. So we looked at these group of neurons called GABAergic interneurons, and there are different classes of them. There's parvalbumin, there's somatostatin, there's calretinin, there are different ones. And they all these GABAergic neurons impinge upon glutamatergic pyramidal neurons, which are the main excitatory neurons in the brain and are involved in connecting neurons and brain regions across large parts of the brain. And this has been shown to be very critical in schizophrenia. There are a variety of GABAergic markers and glutamatergic markers that are disturbed. Bipolar disorder, autism, and major depression also have been associated with abnormalities either of GABA or of glutamate. But they're not the same across these disorders. These abnormalities are different across different disorders. So, and what we found here, I'm just going to show this fairly quickly. This is just to show that the animals, when the pregnant mice were injected at embryonic day 9, which is about early to middle pregnancy, or the equivalent of that, and there was a robust induction of cytokines in the maternal serum. So it showed that this is a drug called poly-IC, so you get much higher cytokine levels. And it didn't affect the maternal or fetal outcomes. So the number of live births and things like that were not altered. So this is a key slide, which is that we looked at a measure of functional connectivity between these parvalbumin neurons and pyramidal cells. And we found that this was decreased in offspring of the mothers that were given poly-IC versus given saline. And this is the poly-IC and this is the saline. So there was this marked effect on functional connectivity of these parvalbumin interneurons. But when we looked at the calretinin interneurons, the ones that haven't been so associated with schizophrenia, we didn't see an association. So it looks like an effect specific to parvalbumin interneurons. And this is a fairly well-replicated finding. In adult patients with chronic schizophrenia and some in first episode. So now we've moved hypotheses. We've gone from the prenatal to adulthood. And the ones that are most effective seem to be things like interferon TNF-alpha, interleukin-6. These are fairly high effect sizes, 0.73 and 0.81. There are also PET studies that I'll show you on neuroinflammation and schizophrenia, and some evidence that anti-inflammatory drugs, like the COX-2 inhibitors, the aspirin even, may decrease certain symptoms in schizophrenia. And the idea is a specific cytokine antagonist may target a common pathophysiologic abnormality in schizophrenia. This is just showing what I've summarized. It's a meta-analysis done by Brian Miller, so it's showing higher cytokines. These are all the cytokines here in schizophrenia cases in this meta-analysis. And this is a model that we proposed in a paper that we published in Biological Psychiatry last year. And the idea is that this is just more of a hypothesis or a model, but the idea is maybe maternal immune activation by increasing cytokines leads to aberrant fetal programming, maybe through epigenetic mechanisms or other mechanisms, and that leads to an increased brain and peripheral cytokine response that could occur in childhood and even in adulthood, leading to more neuroinflammation and also decrease in hippocampal volume. So, and this kind of provides a way to, and I'll show you a way maybe how we can link the animal models with the human studies. But first, these are some imaging studies of inflammation in schizophrenia and autism. So there's this translocator protein called TSPO, and it's localized on microglia. And there are ligands that have been developed, and these ligands, when they bind to them, they bind to activated microglia. And the microglia are the resident immune cells in the central nervous system, including the brain. So this is a way to try to look at neuroinflammation in the brain of live subjects. And there have been some recent ligands that have been developed. The one that's been most recently developed, called a second generation, did show an increase in the binding of this ligand in the frontal and temporal gray matter of ultra-high-risk psychosis patients and in for schizophrenia patients as well. So the ultra-high risk was interesting because these are people who largely have not been medicated, certainly not with antipsychotics. So it doesn't seem to be an effect of the antipsychotic. It might be indigenous to the illness. And these other studies showed varying effects. So it's unclear. There have been some positive studies, some negative studies, but it's a relatively new area of research. So the idea ultimately is how can we go from the lab to the clinic? How do we go from epidemiology to the clinic? One way to do that, and what we might call a personalized medicine approach, is to look at individuals who have high levels of inflammatory biomarkers to target for treatment. And in this study was done by Andrew Miller. Ray Zahn was the first author, published about 2 years ago, where they use infliximab. And infliximab binds to and blocks TNF-alpha. It's an antibody. It's a monoclonal antibody. And they gave it in addition to antidepressants in treatment-resistant depressive cases. And at 12 weeks of follow-up, infliximab was superior to placebo, but only in subjects who had a baseline CRP level of greater than 5, which is pretty high. Although in this study it was about 30% of the sample had that. So this is kind of a proof of concept study. It's a pilot study. It doesn't prove anything, but it suggests that maybe we can be thinking about this not only for depression, but maybe other psychiatric disorders in which inflammation could play a prominent role. So we got some funding from the Stanley Foundation and from Roche to do a trial of a monoclonal antibody to a different cytokine, IL-6, and that's called Actemra or tocilizumab, which blocks the IL-6 receptor. So it's kind of a targeted treatment approach. In our study, we didn't have enough subjects that we could measure, only include people who had inflammation, who had schizophrenia. So we took people who had either moderate or severe schizophrenia symptoms, and these happen to be chronically ill schizophrenia cases. We'd like to do this in prodromal cases, ideally, but we started with the chronically ill. We're currently doing this trial now. We're about halfway through, so we don't have findings to present on it, but I think this is a kind of an interesting, potentially new approach to target the immune system, an activated immune system, to see if it reduces positive symptoms, negative symptoms, and neurocognitive abnormalities that are found in schizophrenia. And it gets us this issue of cause versus correlation. Because if you see a chronic patient with schizophrenia and they have an elevation of cytokines, you don't know if that's a result of the illness or factors that are associated with schizophrenia, or is it a cause? Well, if you can dampen down the cytokine effect, decrease brain inflammation, and the symptoms get better, that gives you pretty good evidence that you have a causal association. And we have now a preclinical study on maternal immune activation and what's called cerebral blood volume. It's kind of like a functional magnetic resonance imaging approach. It's looking at the amount of blood flow in the brain in response to different insults. And there was a study published by Scott Schobel at Columbia where elevated CBV was found in individuals who were prodromal for schizophrenia and who then later converted to get schizophrenia. So it was an important predictive factor, and it was only in the hippocampus that the finding was observed. So the idea of this study in a preclinical study is to give maternal immune activation to the mouse in that same study I showed you earlier, the same type of a study, but now do brain imaging on the mice to see if that causes hyperactivation of activity in the hippocampus. We then want to see if that increases not only early in the mouse's life, the life of the mouse, but also later. Does it lead to a persistent effect? And then finally, we want to see if we give this Actemra or tocilizumab, does it prevent elevations in hippocampal cerebral blood volume? Does that then dampen down what we think might be I can't show you the evidence, but there's evidence that this increase in volume might be reflective of an inflammatory reaction. So we're currently doing that at present, and again, I don't have data to show you, but it's kind of highlights the importance, how one can translate again from the epidemiologic findings here to a preclinical study, and potentially have relevance to treatment. And we're currently doing the same thing in humans who are treated before and after tocilizumab. We have a very small pilot study. But it's kind of nice to be able then to go between the human and the animal with regard to the brain response to immune stimulation or infection and treatment, a treatment effect, a pharmacologic effect. And then in closing, I'd like to cite my many collaborators in this work who work across an array of areas, including birth cohort studies, epidemiology, brain imaging, neuropsychology, animal studies, people in Finland, people in California. It's a cast of hundreds well, at least, the total number of people probably is 30 or 40 people who are involved in this, and my funding sources.